All right, so it is a great pleasure to have Karel Svoboda speaking today. I've known Karel for about, 20 some odd years at this point. Karel did his undergraduate work at Cornell University, where he got a degree in physics. He then went on to Harvard University, where he completed his Ph.D. work in biophysics with Steve Block, where he was really in the vanguard of people using optical trapping to measure the force, the step size, and the velocity of single motor proteins, in this case, kinesin. After his PhD work, he went to Bell Labs, where he worked with Winfried Denk and David Tank. And there he was really, again, in the vanguard of people. Well, in fact, Winfried Denk invented two-photon microscopy, but they were using two-photon in really interesting and new ways, particularly in vivo, to measure dendritic spine compartmentalization and dendritic calcium dynamics in the somatosensory cortex of rats. For this work, he eventually won the Brain Prize, which is the Nobel Prize for Neuroscience. After his postdoctoral work, he went to Cold Spring Harbor Labs, where his work really blew up. He moved from imaging spine compartmentalization and the gain and loss of dendritic spines to measuring, to revealing local circuitry, interareal circuitry, the involvement of different areas during behaviors, and then since left Cold Spring Harbor to move on to Janelia Farms, where he became a member of the National Academy of Sciences and has extended his work now to understand the circuitry of planned movements involving multiple areas, both in the cortex, subcortically, and in the brainstem. And he'll talk about that today, I think. I suppose I'm supposed to, I'm not very good at these introductions, but I can say if you're a graduate student out there and you're listening to this, I would deeply encourage you to apply to Karel's lab. You will learn more in 2 years working with Karel than I think at any time prior to or after that. Karel's also a guy that has just a hilariously good sense of humor with an unbelievably dry delivery. And yeah, if you get the opportunity to work with him, jump on it. He's a fantastic mentor, a great scientist. And speaking for his mentorship, he has mentored dozens and dozens of students who've gone on to their own labs, some of whom are now members of the National Academy and Howard Hughes investigators, and all of whom have been doing fantastic research and really moving the fields that they've joined, moving these fields forward in unprecedented ways. And all of them using the signature Karel Svoboda approach, which is to really measure things with great accuracy and great clarity, and to really drill down on what is the most important question to be asking. I think above all, that's what he really teaches the students. And anyway, that's enough said. I'll let Karel do the talking. It's again, it's a real joy to have him here and a great pleasure. Thank you.
Oh, thanks, Josh. I was a little worried when you were introducing me, but you were remarkably well behaved. That's good. Okay, thanks. That's very generous of you. It's been quite a time since Josh and I worked together and kept in touch. So, thanks for that introduction. So I'll tell you about some work that we've done over the last 5 or 6 years all at Janelia. And it's related to decision-making, motor planning, initiation of movements. Parts of it are published, and I'll go through that stuff relatively quickly. The last bit is on going work, on movement initiation. And generally speaking, the goal in our laboratory is to decipher how neural circuits process information, produce behavior. This is kind of a core theme, of course, of systems neuroscience. And, in our kind of book, Understanding Neural Circuits, rests on 3 pillars. The first is, of course, measurement of neural activity, what systems neuroscientists have been doing for decades, especially in the context of behaviors, and the behaviors define kind of the problem that is being studied. And we'll be talking about decision-making, short-term memory, and the planning and execution of movements. And of course, information in the brain is processed by patterns of action potentials, also referred to as neural activity. And measurements of neural activity provide hypotheses, of course, about how these patterns of action potential represent behavior-related information, how they relate to behavior. The last decade has seen great advances in methods for measurements of large populations of neurons, and of course, methods for manipulating neural activity. And some key advances really have been made at UCLA. Now, neural representations, these patterns of action potentials, really an emergent property of neural circuits. So the second pillar for us has been model. We want to understand how these neural representations come about in neural circuits. So the second pillar is models of neural networks and computation. And generally speaking, these are mathematical models, some long in the tooth, that provide hypotheses about how neural representations come about. And these models are typically based on highly simplified neurons and circuits and sort of encapsulate in some sense the principles of neural dynamics and computation. And for the last 50 years or so, the worlds of experimental and theoretical neuroscience have mainly existed in parallel with little interactions, I would say. And I think these are exciting times because it's becoming possible to have large-scale recording methods, manipulation methods to test long-standing models of neural computation in behaving animals. And we're very excited by this direction. Now, ultimately, we'd like to understand, of course, how biological matter computes. And this really brings me to the third pillar here, which is really about cell types and their connections. And again, it's an exciting time because the field is making great strides in delineating the brain cell types and connections over all length scales in the brain. So this picture shows the motor cortex neurons up here and their projections to a dozen brain areas in the thalamus, the midbrain, the medulla. And one theme in my talk is really about how multiple brain regions, multi-regional circuits interact to perform neural computations. That will come up especially towards the end of the talk. And so today I'd like to tell you about collaborative work on motor planning and movement initiations that rests on all three pillars. So what is motor planning? Well, motor planning is first an important aspect of motor control. It's known that many skilled movements are too rapid for online correction. So here, this is the example is a baseball pitch. And movements that are preceded by a planning process during which movement parameters are set are faster and more accurate than movements that are not prepared or planned. Okay, so movements are faster and more precise when subjects are given time to prepare them. It's sort of mental pre-play of the movement, planning of the movement makes the movements faster and more accurate. Now, motor planning is also a kind of short-term memory that links past events and future movements. It's a kind of a prospective memory. Again, here, the pitching example here, the catcher, the pitcher plants a pitch triggered by the catcher's hand signals, but many seconds might elapse between the catcher's hand signals and the consummation of the pitch. And during this time, the instruction from the catcher and the motor plans are stored in the pitcher's head, in neural activity, in neural circuits of the pitcher's brain. And this is quite mysterious. How are these dynamics that play themselves out over timescales of seconds, internally generated and maintained dynamics, how are they produced by neural circuits? Now in the lab, motor planning is studied generally in what we refer to as delayed response tasks. And these have been around, especially in the context of memory-guided response tasks, especially in awake behaving primates for decades. And we started or developed a delayed response task in mice. So in our experiments here during the, there are kind of behavioral epochs in the experiment. There's a sample epoch during which mice perform a simple sensory discrimination. The details don't matter. These are auditory discrimination experiments, but we've also done tactile discrimination, holds for both cases. And mice then indicate the decision about the sensory stimulus with short latency, skilled directional licking during a response epoch, licking to a target. Okay, it's a short latency responses here. You can see that after a go cue that signals the beginning of a response epoch, the reaction times are only on the order of 60 to 70 milliseconds or so. But mice are trained to respond after delay epoch lasting seconds. Okay. And during the delay epoch, the mouse relies, as the picture, on internally generated maintained brain activity to bridge sensory information during the sample epoch and the action in the future during the response epoch. Now, during the delay epoch, so this is the mouse essentially, the representation, the motor plan can be thought of as a short-term memory because what we know from behavioral experiments is that the mouse actually remembers what to do next. And for that reason, we refer to the short-term memory during the delay epoch as motor planning. Now, as I've already alluded to, behavioral experiments in humans, but also in this particular experiment, show that mice actually use the delay epoch to plan fast and accurate directional licking. So if you shorten the delay epoch, the subsequent movement becomes, the onset of the movement becomes slower and less precise. So in this behavioral experiments, the sample epoch, the delay epoch, and the response epoch really kind of correspond to different aspects of behavior: decision-making during the sample epoch, motor planning, short-term memory during the delay epoch, and movement and action during the response epoch. Now, a key step in the analysis of neural mechanisms, under identifying a key circuit node underlying this behavior, in particular, we started by looking for circuit nodes that underlie motor planning, is a recording of neural activity and inactivation experiments. So we identified over the last, using a variety of methods, over the last 6-7 years, the anterolateral motor cortex as a brain region that actually plays critical roles in this behavior, both in decision-making, movement planning, and also, as I'll show you later, in movement initiation. And this is, these processes are reflected in the activity of ALM neurons that are recorded here with silicon probe electrodes. So what you see here is raster plots. You've probably seen many of these, trials rate in the vertical dimension, time in the horizontal dimension, and below spike rates that correspond to these raster plots. The blue corresponds to right movement trials, red to left movement trials, and they're of course interdigitated in the experiment, but pulled apart here for visualization. You can see here, for example, a neuron that becomes selective during the sample epoch for right movement trials and increases its selectivity during the delay epoch, okay, corresponding to this process. We refer to this also as preparatory activity. This is the neural correlate of motor planning. This neuron becomes even more selective right around the onset of movement in the response epoch, right after the go cue. Here is another neuron that is selective before left movements, okay? And this neuron actually loses selectivity and activity right around the time of movement. Here's a third neuron that becomes, has little selectivity, but is strongly phasically active around the time of movement onset. And here, a fourth neuron that is phasically active around the time of movement onset, but has selectivity for right movements, okay? So the vast majority of neurons in ALM show some sort of task-related activity with this complex dynamics. Now, why did we pick ALM for recording originally? And this was based on an inactivation experiment where we inactivated a bunch of cortical areas, one cortical area about a cubic millimeter in turn during the delay epoch. And we asked a simple question: when inactivating which brain region, is there an effect on the subsequent movements? And we found that that ALM, but not other cortical regions, are critical to plan movements. And what ALM does, ALM inactivation does, it produces a kind of hemimotor neglect. With ALM, unilateral ALM inactivations, mice have trouble planning movements or facial movements in particular into the contralesional space. And this is similar to what is seen with primates and humans with lesions in posterior parietal cortex or premotor cortex, and the same kind of effects we see also with chronic lesions. Okay, so mice have trouble planning movements in the contralesional space after with ALM inactivated. Okay, so both from evidence from recordings that show preparatory activity for specific movements, and inactivation experiments sort of highlight that ALM is involved in motor planning. So on this baseline, we can now dig further into causality and mechanisms. How precisely is the dynamics now of neuronal populations, not just single neurons related to behavior? As I mentioned already, the activity patterns in ALM neurons are diverse, have diverse selectivity patterns and dynamics. How do we relate the activity of neural populations to behavior? Now, recordings from large populations of neurons that have become possible over the last decade or so have enabled state space analysis of neural activity. And you've probably heard about these kinds of tricks to analyze neural populations. And the ideas have been around for decades. They've sort of come into vogue over the last decade because of these large-scale recording methods that provide essentially the statistical firepower to do this properly. So in the state space analysis, we'll analyze spike rates rather than individual spikes. So these are spike rates averaged over tens to 100 milliseconds, depending on what we're doing. And so what we mean by state space analysis is that we think about the recording, simultaneously recorded neurons, so say N neurons, as essentially the recording and the activity patterns tracing out trajectories in an activity space where high dimensional activity space and n-dimensional activity space where each dimension is the activity of one neuron, okay? And so each axis is the activity of each neurons, and the population activity then traces out a trajectory in this activity space. On average, of course, trajectories corresponding to different movement directions trace out different trajectories in this activity space. And then during the delay epoch, this activity evolves relatively slowly and then more rapidly around the time of movement onset. And so and we can then use dimensionality reduction methods to extract interesting features of the data. And I'll first illustrate this for preparatory activity during the delay epoch, so this delay related activity, and later turn to movement related activity during the response epoch. So how do we do dimensionality reduction? There's many ways of doing this, and several methods produce consistent results. There are different ways, of course, of doing dimensionality reduction, and the validation of the approach so what dimensionality reduction does is it essentially highlights particular kind of features of the data, and the particular dimensionality reduction method, there's no magic, nothing very principled about doing this. It's validated in the sense that the dimensionality reduction method actually explains that the activity in particular kind of projections in the activity space explain a lot about the activity itself and also about behavior, okay? So what we have done is defined a choice dimension in activity space, and that's the direction in activity space that best separates the two activity trajectory, and then project the activity along this dimensionality, along this choice direction. This is one aspect of dimensionality reduction. And this is one view of the data that reveals a particular aspect of the data. And it turns out that most of the movement selective activity, the activity during the delay epoch that predicts whether or not the animal will make a right movement or left movement, is contained in this projection, okay? And activity along the choice direction, and we also refer to these directions as modes, as great explanatory power, okay? And it turns out that in these dimensionality reduction tricks, some slices, some views of the data are more useful than others, and that they teach us more about how activity relates to behavior or how patterns of activity are generated. And this trick is particularly useful in this context. So what you see already is that population activity in delay shows sustained activity patterns during the delay epoch, where this is now a you can think of this as a multi-neuron peristimulus time histogram that extracts the selective activity from the neural population during the delay epoch. And you see that there's sustained activity and selectivity related to upcoming movements lasting seconds. And to some extent, this is by construction. And it turns out that on a trial by trial level, okay, activity in this projection actually predicts behavior. And I'll show you examples of that. And this activity and selectivity is internally maintained by the brain over timescales of seconds, right? The duration is set by the experimenter, is related to the duration of the delay epoch. And so the next question is, how is this activity maintained in the brain? As you may know, it's not trivial for neurons to maintain persistent or slowly varying activity like this preparatory activity just I've shown you. This is activity during the delay epoch over timescales of seconds. And the reason is that neurons have time constants on the order of tenths of milliseconds. Okay, so if you inject current or information into isolated neurons, the information or the excitation decays over timescales of milliseconds. So the way to think about it is only one fixed point in the system close to rest. So what that means is that neurons in isolation are more or less memoryless and can't maintain preparatory activity. And we've actually checked this in the intact brain by doing whole cell patch clamp recording. There's nothing special about ALM neurons that show this persistent preparatory activity over timescales of seconds, okay? And so persistent preparatory activity, such as the activity pattern seen right here in ALM, is an emergent property of neural circuits. In particular, the time constant, of course, of the response of individual neurons can be prolonged by implementing positive feedback. For example, using a circuit motif that we know exists within ALM. So for example, neurons that predict contralateral movements, here in blue, or right movements, if you're in the left ALM, are more strongly connected to neurons that, to other neurons that have similar selectivity, both similar kind of dynamics that are selective at similar times and that show similar direction selectivity. This is a recent paper we published in Nature Neuroscience if you'd like to see more of that. Okay, so one neuron excites another. So as the activity in one neuron dissipates, right, it gets a kickoff excitation from the second neuron, prolonging the network time constant. And with the appropriate pattern of appropriate pattern of feedback, you can achieve persistent states. So here now, for example, in this cartoon here, two persistent states. And what these things are referred to are fixed points of the system, where the activity, if it reaches the population activity reaches the state, it is maintained. And at that point, dissipation and feedback are balanced and activity states are maintained. And this particular kind of system is also referred to as a discrete attractor network. And here, each of these stable states correspond to what is called fixed points. And this system has two fixed points, one at 0 and 1. And in this kind of discrete attractor network with discrete fixed points, the hallmark is that small deviations of activity away from the fixed points will cause changes in activity that tend to dissipate the deviations and move the network back to the fixed points. And what I'd like to show you in the next couple of slides very quickly is that memory-related activity in ALM corresponds to fixed points in discrete attractors, but with a couple of twists, okay? So what are these twists? So as I mentioned earlier, so fixed point corresponds to stable activity pattern viewed classically. But in general, as I've shown you at the level of individual neurons and also at the level of populations, ALM neurons and populations ramp, so shows slow drifts in activity over time, right? And so the question is, how is this ramping activity consistent with fixed points in a discrete attractor network? Well, what we think is happening is that the activity sits at a fixed point, but the fixed point moves in activity space over time. This is, again, one of these trajectories after a dimensionality reduction for correct right movement trials. Okay, you see this kind of a ramping drift for this particular ensemble of neurons. And what happens is that the fixed point moves in activity over time. The activity sits at the fixed point, but it's the fixed point that moves, okay? To directly show that the activity trajectory actually corresponds to moving fixed points, we need to do a perturbation experiment, okay? And so, the logic is simple here, okay? So basically, we perturb the system at some time. After the perturbation, the perturbation, of course, leads to a distortion of the activity trajectory for a discrete attractor system. We expect that after the perturbation, the system recovers, okay, to the fixed point. So the trajectory should recover rapidly to the fixed points corresponding to the unperturbed trajectory, the dashed line, and then continue along the unperturbed trajectory as the fixed point moves. So let's look at the experiment. And I now regret not adding a bit of detail about the inactivation experiments here, because a lot of the trickery, the way how to get these experiments to work is really to figure out how to perform an experiment, a perturbation that distorts an optogenetic perturbation here that distorts the trajectory sufficiently, that has some behavioral effects, but doesn't break the behavior. Those are the most insightful experiments, so we can study the recovery and the response of the trajectories to perturbation without the whole system being messed up. And this is something that this is a lot of work that was performed by Hidehiko Inagaki, who got these experiments to work. And what we're basically doing here is an ALM bilateral transient inactivation by driving GABAergic interneurons, but at a kind of moderate level that transiently collapses selectivity, and so that the trajectory recovers to the correct trajectory on a subset of trials and to the incorrect trajectory in another subset of trials, which then is associated with behavioral errors. And so here's an example of a set of experiments where the animal made the correct choice, which we can already tell by the shape of the trajectory right here. We then apply this optogenetic perturbation, distorting the trajectory. And what we find is that on correct trials where the behavior is not changed by the photostimulation, okay, the activity rapidly snaps back to the unperturbed trajectory here, the dashed line. And in particular, that rate of recovery is much faster than the rate of ramping. This is because the trajectory, because the particle rolls down the sides of the energy well before the fixed point can move a lot. Let me also mention, I forgot to mention that I should have mentioned this earlier here, that there are different ways to look at attractors. One of them is an activity trajectories. The sort of a classic representation is also in the context of particles in energy wells, of course, where this is now the particular projection in activity space that we're studying, and the state of the system for one of the two movement directions, okay, in this representation. Okay, so here again, the attractor corresponds to a particle in an energy well. Okay, where were we here? Okay, so this is on correct trials, okay? And this and other data provide support for the discrete attractor model. And it argues also against other models. So for example, there are other kinds of attractor models that have discrete attractors, but something like continuous attractors are very common and popular in the modeling literature. And you know, this is what you'd expect, the kind of recovery you'd expect for line attractors. So it's really the rapid recovery to unperturbed trajectory is consistent with the discrete attractor. Moreover, what we find is that multiple fixed points coexist during the delay epoch. Okay, and here this point that I made in passing of producing errors in behavior with perturbations is key. Okay, so here and notice some other kind of features of the perturbation that transiently during the perturbation trajectories for right movement trials or left movement trials are undistinguishable. Okay, and on correct trials, the right movement trajectories snap back to the correct right movement trajectories with correct behavior. And on left movement trials, the left movement trajectories snap back to close to the left movement trajectories, again with correct behaviors. On error trials, behavioral error trials, the trajectories are reversed. Now, the left movement trajectory overlies the right movement trajectory, and the animal makes a directional movement error, and vice versa. Okay, so in the energy picture, what is happening here is that the perturbation brings the network close to the separatrix, right? The kind of the bump separating the two fixed points, okay? And on error trials, and on correct trials, the system recovers to the correct fixed points. On error trials, the system now decays in the incorrect fixed point with an error, with a behavioral error. And so together, these experiments provide strong evidence for discrete attractor dynamics underlying preparatory activity. Now, based on these types of experiments and modeling, we arrived at the following picture underlying decision-making and motor planning. So during the task, an external input to ALM, and it's not just ALM, but it's a cortico-thalamocortical loop that maintains and generates preparatory activity. An external input to the system triggers a transition from one fixed point, okay, kind of a simple attractor with one fixed point, to a system with two fixed points. And of course, this is learned. This is only for this two-choice task. For a more complex situation, there'd be more fixed points. And this is also referred to as a pitchfork bifurcation in the dynamical systems literature. And the sensory input into ALM from auditory cortex or somatosensory cortex, depending on the modality, biases occupancy to one or the other of the fixed points. So here, the system is biased towards right movements because the sensory instruction biases, instructs right movements. And what happens over time is that the external input pushes these attractors apart and also makes these attractors of fixed points deeper. Okay, and this ramping, this pushing apart of the attractors is related to basal ganglia loops, we believe. This is ongoing work that are also in the systems neuroscience literature. This is also related to the phenomenon related to urgency. If you, for example, manipulate the duration of the delay epoch, the ramping becomes steeper for shorter delay epochs and less steep for longer delay epoch. And this is a basal ganglia loop is critical to generate, to push these attractors, to generate these attractors and to push these attractors apart over time. Now this has this pushing apart over time and a deepening of the attractors has two important consequences. First, as the attractors move apart and become deeper, it produces robust preparatory activity and behavior. That means that activity patterns later in the behavior becomes insensitive to input from other areas because the system sits in deeper attractors and the attractors are further apart. So along this activity axis, you have to provide a much larger perturbation to switch the behavioral outcome, okay? And it turns out that this mechanism implements gating of signal flow between brain regions during behavior. This is kind of an exciting corollary that I won't go into great detail, but just sort of highlight it in one slide. We recently put a bioarchive up on that you can have a look at. So for example, a brain region like sensory cortex can of course influences the target brain region, in this case ALM, greatly early in the sample epoch, because it determines which activity trajectory is chosen, right? Either right movement trials or left movement trials. So there's a huge effect early on. When the same sensory stimulus comes in late during the delay epoch, the perturbation is just not strong enough to change change the trajectories and the perturbation from sensory cortex decays. And I think this is kind of an exciting corollary, because a dynamic attractor landscape like this could be a general mechanism that underlies flexible routing of information across brain regions, right? At some point, there's strong influence from the sensory cortex over motor cortex. At some other point, the influence is weak, okay? So, and it really depends on what state ALM with this sort of the ALM thalamocortical circuit is. That determines what the influence of sensory cortex over that system is. And by the way, if you can read more about this study in this preprint and also in Anne's really nice, actually James Roach and Anne wrote up a blog post about that study. Thanks, Anne and James. Okay, so let me finish this part up with some just quick remarks. So it's super exciting for me that we can now test long-standing models of neurodynamics, for example, neurodynamics that underlie motor planning and short-term memory. I was at Bell Labs, as Josh mentioned, in the 1990s, and we worked on two-photon microscopy and cellular stuff, but there was a lot of buzz about attractor networks, and these models were invented there, most interesting models, for example, by John Hopfield. And back then, it was difficult to see a path to directly testing or disambiguating between different network models. And what has changed is that we now have, of course, quantitative behavior in mice, where it's possible to do experiments at reasonable throughput. This is actually quite important. Lots of experiments required for this kind of stuff, as some of you know. And measurements from large numbers of neurons allows us to characterize the neural dynamics in activity space reasonably thoroughly. And dimensionality reduction methods allow us to then extract the relevant features of the dynamics. In this case, of course, relatively low dimensional, because it's a simple behavior that has helped us here, but I think it revealed principles of how persistent preparatory activity is maintained and how brain regions communicate and so on and so forth. And importantly, optogenetics allow us to shape neural trajectories and watch their recovery. And I think it's really the dynamics of perturbed trajectories that discriminates between models. There are, of course, many open questions, how, for example, how attractor landscapes shaped by learning to support skilled movements, how are attractor landscapes shaped in more natural behavior. what are the mechanisms underlying short-term memory involving continuous rather than discrete variables, right? Like a pitch of a sound rather than discrete movement directions. There's a lot, and we're working on some of these problems. It's, for example, possible that continuous functions are approximated by a multitude of discrete attractors. Interesting question. And yeah, that's pretty much it for that. All right, let me now turn to movement initiation and go back to the basic behavior. So we have here in this behavior triggered by an auditory go cue, mice respond with, as I mentioned, with rapid skilled directional licking. So this is the first lick in about and a later lick in about. And I should say that this rhythmic licking is controlled by brainstem circuitry in the medulla, where there's a CPG that drives the orofacial movement under water consumption. And this happens, this onset of this bout of licking happens over times with reaction times on the order of 60 or 70 milliseconds. Okay, so the question is, what are the neural mechanisms that convert a motor plan into the appropriate movement at the appropriate time? And it's, I think, behavior in some sense is all about movement. And there really is no holistic description of how movements are initiated at the level of multi-regional circuits. And we wanted to take a try, at least in this relatively simple cued movement context, to try to understand how a motor plan is converted into a movement, where the movement happens, where the right movements has to happen at a particular time. It's the ongoing work of Susu Chen and Hidehiko Inagaki, and in their native habitats in the laboratory. So Hidehiko moved to the Max Planck Florida Institute recently, also a terrific person, starting up his own lab there. And so now moving beyond preparatory activity during the delay epoch, we're going to do similar kind of state space. We're going to start with similar kind of state space analysis during the peri-movement epoch. And again, following the footsteps, in this case, of folks who've worked in motor control like Krishna Shenoy and Mark Churchland and others. And as you can already see, neural activity is modulated quickly, is very dynamic around the time of movement onset, and is sort of highlighted here by this rapidly changing squiggly line after the end of the delay epoch. Now, again, we use targeted dimensionality reduction to extract interesting feature of the data. And during the delay epoch, during the response epoch, like during the delay epoch, we extract what we refer to as a motor command mode. Okay, now here, the motor planning mode during the delay epoch extracted as before. Here's the motor command mode extracted during the peri-movement epoch. We again look for the direction and activity space that best separates activity correlating with right movements and left movements, or contraversive movements and ipsi-versive movement. And as highlighted by others, for example, Matt Kaufman et al., this direction and activity space is orthogonal to the motor planning mode, kind of lends support to the notion that during planning, cortical activity exists in sort of one particular kind of output null space, preparing a movement without moving, and then switches after the go cue to a motor command mode that is potent for movement. And notice also that very rapidly after the go cue, the motor command arises with the collapse of these attractors that define the motor planning mode. Okay, and similar changes in neurodynamics around the time of movement onset have been noted in other species and behavioral tasks, in particular in primates. So here's, for example, a memory-guided saccade task, selectivity associated with eye movements, preparatory activity, and here the work I referred to earlier from Matt Kaufman et al., where they have, this is now representation across 2 dimensions in activity space, where preparatory activity before movement onset evolves in one direction, and then movement onset is characterized by activity in an orthogonal dimension, where this notion of preparatory activity that is orthogonal to movement potent activity comes from, okay? So now, so this is quite general. This is arm movement, eye movement, very similar kinds of patterns seen as in tongue movement in the mouse. So third activity mode plays a critical role in movement initiation that I also have to introduce. And we refer to this as go-cue mode. And in the literature, it's been also referred to as condition invariant signal. And it's really it's defined by the direction in activity space that shows the largest change in activity with respect to the go-cue. Okay, differences between delay activity and peri-movement activity. And activity along the go-cue mode is non-selective, and the direction of the go-cue mode is also orthogonal to the motor command mode and the motor planning mode. And remember from the single cell PSDHs that individual neurons can contribute to all of these activity modes with different weightings, right? So individual neurons contribute to all modes with different weightings. And so an analysis of latencies shows that the motor command mode actually precedes the reaction time by 20 milliseconds or so. That's good. It could be then that the motor command mode in or the motor command mode in ALM is indeed a motor command that is causally related to the onset of movement. And importantly, the go-cue mode in turn precedes the motor command mode. And it's these go-cue trigger changes in activity, the go-cue mode followed by the motor command mode, that might underlie the initiation of movement. And what I'll show you in the next few slides is that the go-cue mode and the motor command mode, in fact, are dissociable, okay? And that the go-cue mode causes the transition of the motor planning mode, from the motor planning mode into the motor command mode, which then initiates directional licking, okay? Moreover, okay, these modes and functions map on distinct multi-regional circuits. We're trying to bring this dynamical systems view of neural circuits together with actual anatomical substrates. That's what we're going to do in the next few slides. And in particular, to give you a preview, the go-cue mode is initiated by an auditory signal that dribbles into the midbrain, okay, and then propagates through the pedunculopontine nucleus and the midbrain reticular nucleus that should also be in here via the thalamus to ALM, which then again collapses the motor planning mode and triggers the motor command mode, which then initiates movement via descending projections from ALM, which after all is a motor cortex, to the medulla, where these command signals initiate the relevant movement. Okay, so I'll start with the circuits that carry the motor command to put some meat on this. And so here are descending neurons from ALM. So Mike Econom and Sarada Vishwanathan a few years ago discovered a set of neurons. We refer to them as PT lower cells, pyramidal tract lower cells, that send axons to premotor centers in the medulla, including premotor centers that control orofacial movements. It's these magenta neurons. And these magenta neurons, these PT lower cells, actually are enriched in activity related to the motor command, like strong modulation and selectivity just around the time of movement onset, as illustrated here with these two sample neurons. We can now do optogenetic experiments and inactivate these neurons around the go-cue. We do this with chloride conductances that are light gated just for the small number of neurons, small proportion of neurons in layer 5B that project to the medulla. When we silence these neurons right around the time of the go cue and movement onset, the animal fails to respond. Okay, so suggesting that, that input from these neurons to the medulla is critical for movement initiation. Now we combine this with recordings in the ALM. So these are the output neurons of the ALM. What does this do to ALM dynamics? And it turns out that silencing PD lower neurons has subtle but highly specific effects on population activity. So here are the motor planning mode, the motor command mode, the GOKI mode under control conditions in this experiment. And then with silencing the PD lower neurons, the output neurons from the ALM, motor planning mode completely unaffected, GOKI mode completely unaffected as far as we can tell, but there is a selective deficit in the motor command mode, particularly for contraversive movements. And we know that these descending neurons really control contraversive movements. There's an asymmetry between two hemispheres of which movement directions are controlled by these descending neurons. And this is also, it tells us that the GOKI mode and the motor command mode are dissociable because there's an effect on the motor command mode without an effect on the GOKI mode. And it suggests also that the GOKI mode is upstream of the motor command mode. So this non-selective GOKI mode is upstream of the motor command mode, which is also supported by, of course, the fact that the latency of the GOKI mode is shorter than that of the motor command mode. Okay, let's now look for the antecedents of the GOKI mode. So we search for the origin of the GOKI mode. This is ultimately has to be triggered has to be coupled to an auditory signal. And remember that the latency of the GOKI mode is very short in ALM, just 30 or so milliseconds. Okay, so we're looking for signals that might drive the GOKI mode throughout the brain that have very short latency. So I'll show you the latency analysis across the brain. And these experiments were enabled by large-scale recording experiments using neuropixels probes. So we do multi-regional recordings. We then recover where the electrodes were and by extension where the recorded neurons were. And that's combined with anatomy that I don't have time to show you. And here's the latency analysis across brain areas, okay? It's a complex slide that summarizes really a couple of years of data collecting. And what we find is that the GOKI mode is initiated by auditory information that comes in to the midbrain through the lateral nucleus of the lateral lemniscus. This is an auditory nucleus. And at that point, the auditory responses are non-specific. The nucleus of lateral lemniscus provides input to the pedunculopontine nucleus and also parts of the midbrain reticular nucleus. So this is not limited to any one, this function is not limited to any one midbrain nucleus. It's a couple areas close to the pedunculopontine nucleus. And starting the pedunculopontine nucleus, it's a GOKI signal in the sense that the responses are specific to the learned GOKI sounds, response to the GOKI sound, but not to other control sounds, okay? And then from the latency analysis, you can see that the pedunculopontine nucleus is the shortest latency in showing these GOKI mode responses, just a couple of milliseconds slower than the pedunculopontine nucleus. And then the signal spreads across various brain regions in the midbrain and the hindbrain, and in particular travels into the thalamus that projects to motor thalamus that projects to the ALM here, summarized as thal ALM, which includes parts of MD and parts of the VM. And from many experiments, we know this is the critical pathway for the GOQ signal to reach ALM. In particular, of course, we now hypothesize that the GOQ signal ascends from PP and MRN through the thalamus into the ALM. And then to test this hypothesis, again, we do optogenetic experiments. And because the GOQ signal is a phasic response, non-selective, we can mimic it with channel-rhodopsin manipulation. We now replace in a subset of trials the GOQ with stimulation of channel-rhodopsin positive axons coming from PP and MRN into the thalamus. We excite these axons in the thalamus. And then we do this under conditions where only a small proportion of trials cause a behavioral response. This is more informative and for practical reasons better. So in the absence of a no-GOQ, there are few behavioral responses. When we now add the photostimulus, there's a substantially larger number of responses by 10 to 20%, right? And importantly, okay, so it's a relatively weak stimulus, but importantly, when the animal responds, it responds in the correct direction. Okay, so if anything, the behavior actually improves. We don't quite understand this because it's actually statistically significant. But even with unilateral stimulation of the midbrain axons, we get responses in the with the appropriate directional response. So this is not just some kind of directional startle signal. The behavioral responses are consistent with the sensory instruction. I notice I'm running out of time, but I'm also almost done. And so just show you the most important slide now. This is the recording during this manipulation. And it turns out that the photostimulation also produces the large reorganization of thalamocortical activity. Okay, so just very quickly, control conditions, again, the three modes, GOQ omitted, the motor planning mode persists, the motor command mode and the GOQ mode, of course, don't develop. On stimuli that don't cause responses, the motor planning mode persists, motor command mode doesn't develop, GOQ mode doesn't develop. On stimuli, photostimuli with responses, the selectivity, the motor planning mode collapses, the motor command mode develops almost normally, and the GOQ mode is normal. And the converse is also true. We can silence these axons going into the thalamus and abolish behavior. Just very briefly summarizing. So the midbrain carries short latency GOQ signals through the thalamus into ALM. The signal is required for cued movement initiation, and this acute movement is critical for to convert movement planning activity in thalamocortical circuits to a motor command and release the appropriate movement. And finally, I want to just highlight that the midbrain is, we took this ALM-centric view of movement initiation because that's where we started off with, but the midbrain, these midbrain regions also project to the other parts of the brain that are known to be involved in cued movement initiation. And so I think these midbrain structures are perhaps perfectly poised to coherently control brain-wide circuits underlying cued movements. All right, let me highlight and thank the folks who've done these experiments, in particular the recent work that's ongoing, spearheaded by Hidehiko Inagaki and Susu Chen. I've already mentioned them earlier. Zenkai Go Noli started the work on motor preparation in the laboratory now 10 years ago or more. I've shown you also work from Sandro, done in collaboration with Sandro Romani, Arseny Finkelstein, and others. And Tim Harris and group and Marius Bhattacharya helped with matters of spike sorting and analysis. And finally, I want to very quickly mention that we'll be moving from Janelia, one fantastic place, to another, to an institute later on in the year. And if you have any interest in team science in the context of neural computation in multi-regional circuits, in defined neural circuits, please get in touch. Thank you. I'd be happy to take questions.
Do you want to do it, Karel, where they just unmute themselves and ask the question directly rather than going through me?
I would say, please ask.
There's questions in the Q&A. They won't have the ability to unmute themselves.
Oh, they can't unmute themselves.
They can't. No, that we have all the questions directed in the Q&A.
Yeah. Okay. So let me answer some questions here. So how stable are these delay-related ramping patterns across days? This is a question asked by Payman. These, generally speaking, that depends on cell type. And in general, in superficial neurons, this is often then done with imaging. I didn't show any imaging data. In superficial neurons, these activity patterns are quite stable, at least in trained animals. They are strongly modulated also by the state of the animal. So as an animal becomes sated, generally speaking, the urgency signal becomes less pronounced and the ramping shallower. So even within a session, there is some modulation that relates to the state of the animal. Let's see. And might at least some of the neurons that reflect the go mode be driven by the sensory cue? Is the go mode ever modulated by amplitude or frequency of the tone? So I'm not sure if I answered that, Anne. So this is a question by Anne Churchland. The go mode is in fact triggered by auditory input. It's the nucleus of the lateral lemniscus is in fact an auditory signal. It has to be auditory, but once it reaches the peduncular, the PPN and midbrain reticular nucleus, as far as we can tell, it's already highly specific to the go cue sound. So it's not just an auditory signal. In addition, it tends to be all or none. And I went over this quickly. This is clearest in the photostimulation data. So either the go cue mode happens or it doesn't matter. So there has to be some amplification somewhere there in the midbrain circuits, and we don't have a handle on that. So it's not, there's some computation downstream of auditory signal that's interesting and non-trivial, but I don't have much more to say about that. Let's see. Okay, so sometimes we find very strong population representations correlating to behavior, but inhibition of these populations has little effect on behavior. We have also seen the opposite, where the representations are weaker, but essential for behavior. Curious what you think about this type of dissociation. This is again a question by Payman. Sorry, there are others who ask questions. I'm just going down the list here. I completely agree. There are, especially once you have trained animals and animals that show behavior-related movements, you in fact see correlations with behavior virtually anywhere in the brain. And that's why the inactivation and manipulation experiments are critical. And as you probably, many of you have thought about, even those experiments are difficult to interpret. So there's a lot of stories about quote unquote off-target effects of loss of function experiments. That's why I think the most exciting experiments is where you do kind of a dynamic perturbation together with a recording, and where you then interpret, where you can interpret the often unexpected consequences of the perturbation with behavior. And in the context of the model, if all of it makes sense, you get slowly a sense of what the brain region might be doing, or the network that your brain region is part of might be doing. That's how I think about this. Dean Bonamano has a question here. The speed of the snapback of activity in the delay period after perturbation seems to suggest that the snapback is driven by input from another area. Is the suggestion that ALM is actually implementing the attractor or reading out the dynamics in other areas? So what we, so you don't need, in a discrete attractor network, you don't need input from another area, right? I mean, but we know that ALM activity, for example, depends on input from the thalamus, and thalamus activity and selectivity depends on input on ALM. And more recently, and this is work really from Zhenkai Guo at Tsinghua, it's becoming clear that that bifurcation is really driven by, again, also input coming in through the thalamus via the substantia nigra reticulata. So you really have to think about a multi-regional network, but I don't think that the snapback, the snapping back inherently requires input from another area, unless I'm missing something. I'd be happy to hear more about this if you drop me an email or follow-up question. There's James Roach. James, thanks for the write-up. Are there stereotyped movements during the delay epoch of the task, and could these drive the ramping input to the ALM through the proprioception? So there are stereotyped movements, and and it could be that movement in some sense is in the loop, so we've done experiments where we, it's a motor cortex area that receives input from somatosensory areas that not so much in the facial system, but for skeletal muscle that have proprioceptive input to the motor cortex that might contribute, but is not certainly the only thing. And of course, the more important issue is really about whether or not the activity so the motor system, the fact that you see coupling to the motor system is perhaps not so surprising, right? Because almost every stage of the brain is just a couple of synapses away from the motor system. And so I think the key experiment are these manipulation experiments, right? If you manipulate ALM activity and you look what happens to the trajectories, you can predict what the animal will do. I've just talked about choices right now, but you can actually predict what the animal will do in detail. Actually, the trajectory of the tongue, the speed of the movement. There are even kinematics that is reflected in the dynamics. So it's what that tells you is that ALM is part of a multi-regional loop, at least, that is critical for planning movements. Whether or not proprioception in some sense is part of that loop, I don't know. One would have to block proprioceptive sensors and compare. Let's see. Alex Kostyuk is asking, how much is known about where the ramping activity that separates attractives comes from, and could the source be targeted with optogenetics? Yeah, so this is not my work, and in fact, the SNR is a key source. So basal ganglia, so this is basically cortex striatum to the SNR, which then projects back through the thalamus into the system. And there is also work from primates. The gosh, the folks from Montreal, forgot his Shistek. They have a similar hypothesis, and they refer to this as an urgency signal. Also have evidence that this arises in the basal ganglia. I'm going to go to some of the shorter questions, or maybe a longer one here. If not mistaken, in a motivational state space approach, the organization of the behavior is dependent on the direction of the sensory stimuli, as well as a whole host of other factors, including biological significance of the goal. In this case, there may be a right bias. That bias seems to be biologically inherent to the consummation linking on the right. What would you predict happen to the preparatory signal if the goal's biological significance would change? For example, if the linking solution would contain citric acid in preparation for acid would trigger more saliva to dilute the harmful acid. That's a good question. I don't have a prediction. I'll have to think about it. Good question. I'm going to make a note of this here. Let's see. The interpretation of the perturbation experiment seems to assume that the perturbation does not influence the energy landscape, but merely the state of the system. On the other hand, you stated that the sensory input can shape the energy landscape. Can you clarify this apparent contradiction? So I'm not saying that the sensory input shapes the energy landscape, but that it's an input from the basal ganglia, and this urgency is related to the task structure, okay, to the timing of the task. That shapes the energy landscape. And then the energy landscape is also shaped through this urgency signal, through satiety. The energy landscape becomes flatter as the animal becomes satiated. So I don't think there is an apparent contradiction. If there is, please follow up. Is the ALM the anterior M1 cortex? So the ALM is the anterior lateral motor cortex is defined by mapping experiments, both activity and by loss of function experiments. I didn't show you the mapping experiments for activity. I showed you very briefly loss of function experiment. So it's operationally defined, and it's at the border of what in the atlas is M1 and M2, sort of orofacial M1 and M2 more medially. And I have no idea who drew those boundaries or why, frankly. But if someone knows, please let me know. So it's centered on, in C57 black 6 mice, 2.5 millimeters anterior and 1.5 millimeters lateral. Okay, does the hippocampus play any role with movement disorders correlated to study of the ALM cortex? We haven't looked much. This is from Sami Yanikian. And last question maybe from Anne Churchland. Also referring back to the first part of the talk, do you think that our understanding of how neurons implement attractor dynamics will benefit from identifying cell types while recording? That's a good question. And the whole question of cell type, why do we get away at all with ignoring cell types? That's perhaps the first question. And the way I think about it is that we've done a bit of cell type specific recording, especially in the context of projection neurons and mostly excitatory neurons. And we find that there are cell type specific differences, but they don't explain a huge amount. And I think that's a reflection of the fact that these different excitatory neuron populations are strongly coupled, right? In the motor cortex, you have essentially superficial neurons that tend to have more sensory-related signals in the motor cortex that feed into a lower layer IT neurons, which then feed into pyramidal tract neurons that descend, those are the neurons that descend out to other brain regions, but everyone is strongly coupled to everyone else. So through excitatory connections, there's this downward direction to the flow of information, but there's strong coupling between these neuronal populations, and that's why you see sort of similar stuff in different cell types. Now, I would predict, for example, that we would, and these are the kinds of experiments that we're pushing, that there's something special about neurons that project to the thalamus, that receive input back from the thalamus in a disynaptic loop. And these disynaptic loops exist and could really be key for implementing the recurrent excitation that is required for these attractor networks. But that's really a direction the whole field has to go to be able to record in a cell type specific manner routinely in these kinds of and more complex experiments. If a last question here, Manuel Butrigo Blanco, if a perturbation to ALM became permanent, is the loss of its function redistributed across other neurons within ALM? So we haven't done, sorry, this now. Yeah, so the question is, the answer is that we haven't done that experiment. We have lesioned ALM. When we do bilateral lesion of ALM, animals never recover in the context of this task. They cannot do memory-guided directional licking. When we do unilateral manipulations, the animals retain lesions. The animals have a contralesional neglect phenomenon, but they learn how to do a good approximation of the task using one hemisphere. And that's the extent of what we've done. We have not gone and sort of lesioned subsets of cells or something like that. Yeah, so let's call it a day, maybe.
Yeah, Karel,
yeah.
That's great. I'd like to thank you for a fabulous talk, and hope we can get you next time in reality.
Thank you, Larry. Thanks for the invitation, and thanks for the excellent questions. And there are a couple questions I'm still studying, but if I haven't answered them, please feel free to follow up with an email.
All right. Thanks very much, Karel. Take care.
Okay. Bye-bye.